I'm Yi-Chen. I'm first-year postdoc fellow at the Schmidt Center. We are delighted to have Dr. Anshul Kundaje for our next speaker. Dr. Kundaje is associate professor of genetics and computer science at Stanford University. His research focuses on developing machine learning and computational framework for regulatory genomics. His work has transformed our understanding of regulatory element, regulatory interaction, and genetic variation in general. Before joining Stanford, Dr. Kundaje led the computational effort of the INCO project and the Roadmap Epigenomic project. His contribution has been recognized with numerous honors, including the Hugo Chen Award of Excellence, the NIH Director's New Innovator Award, and many else. So please join me to welcome Dr. Kundaje.
All right, so I just want to start by thanking Caroline and the organizers for this really nice meeting. I'm excited to tell you about how we've been using base resolution deep learning models to decipher syntax of regulatory DNA and non-coding genetic variation. So hopefully this bridges some of the themes you've seen with the molecular data and genetics using machine learning models. So just to orient us, yesterday, Mark gave a really nice overview of how genome-wide association studies have identified thousands of loci associated with diverse traits and diseases. And as you know, a vast majority of these loci end up being non-coding. So they are not disrupting protein coding sequence, but lie in the non-coding genome. They're highly enriched in regulatory DNA elements. But just knowing that is not sufficient because it's still very hard to pinpoint the precise causal variants, the causal genes, cell types of action, due to various reasons. So this is a major challenge of trying to identify causal effects and mechanism. If you move to the rare disease case, again, you have huge catalogs of rare and de novo variants now from whole genome sequencing of patients with rare disease. And again, there's a large number of variants of uncertain significance that actually end up being non-coding. So to address this challenge, I think you need at least two components. First, you need to profile regulatory DNA. You need to actually understand which parts of the genome are involved in gene regulation. And this is a very multimodal problem because there isn't a single assay that gives you all information about regulatory DNA. So people use a battery of assays to profile regulatory activity. So for example, you can perform ChIP-seq experiments for hundreds of transcription factors in some cell line or cell type of interest, or you can perform attack-seq, single-cell attack-seq, DNA-seq experiments that will give you chromatin accessibility landscapes, or you can perform other kinds of experiments, ProCap and histone ChIP-seq and so forth. So it's extremely multimodal data. With these data sets, we can start mapping regulatory DNA, but if you really want to understand how a variant might affect activity, you do need to understand how DNA sequence encodes this activity patterns. So what we're interested in trying to use this data to decipher, how the DNA sequence encodes this sort of function. And for that, over the last few years, we've built these class of base resolution deep learning models. The basic idea here is you're building a supervised model that's taking sequence as its input and trying to predict literally coverage profiles across the genome at single base pair resolution. So no data processing at all. It's literally taking the raw coverage profiles and trying to map sequence to coverage profiles. And throughout this talk today, I'm going to focus on what I'll refer to as short context models. These are models that will only capture about 2 kb to 5 kb of DNA sequence. Obviously, this is a simplifying assumption. The genome is vast. There's lots of long-range interactions. And ideally, we would want genome-scale models. And there are genome-scale models. But I will show you that many of them actually do not outperform local models. So our goal, sort of from a purely design perspective, is to first conquer the short context zone of models. And they will not be as predictive or accurate as the ultimate long context versions, but if you can't conquer the short context, you probably don't have much hope of going long context. And we've built a series of these models corresponding to various kinds of readouts. And one nice thing is we've converged on an architecture that's a common backbone for all our models. So we don't do as much architecture engineering. We've done that for a few years, but we ended up with an architecture that may not be the ultimate delta optimal, like you get an epsilon improvement in performance, but it's extremely stable. And we've done lots of tests on it in terms of interpretation stability, all of those important aspects. And these models really work beautifully out of the box. So the first model we built was in 2021 called BPNet. This was designed for transcription factor data at base pair resolution for chip exo, chip seq, and cut and run. We recently put out Chrome BPNet, which is a similar model that's designed for tag-seq, DNA-seq, and pseudo-bulk single-cell attack-seq. We have ProCapNet, which is designed for nascent transcription and TSS assays like ProCap, Cage, and Rampage. And we have upcoming assays for upcoming models for report assays, histone modifications, so on. So we're building a series of models that are really specialized for each readout. And I'll explain to you why you just can't take the architecture and just apply it, because it seems like it should just work out of the box. It takes us about 6 to 8 months to optimize any model for any specific readout, and I'll explain why. That's really because of the assay-specific technical artifacts and biases. Once we have these models, we can use them to do exciting things, like we can take the predictions of the model for any sequence, genomic or synthetic. We can backprop to the model. We can use a variety of different feature attribution methods to infer for any query DNA sequence for a specific readout in a specific cell type, what are the base pairs that are likely driving activities. This gives us a very high resolution view of the underlying nucleotides that regulate activity. And again, this is very context specific. So if you take the same sequence and push it through a model that's trained in a different cell type or a different readout, it'll give you a different interpretation of the exact same DNA sequence. You can do this for every sequence in the genome. Our models are small, hence we can scale them for interpretation. And then let's say you run this model over the entire genome and you get these kinds of attribution maps that tell you from the perspective of the model which base pairs are important or millions of DNA sequences. You can then identify regions of high importance and then do very rapid alignment and clustering to summarize or distill out motif patterns, recurrent motif patterns that sort of capture the lexicon that the model has learned for predicting activity. Then you can do more interesting experiments. This is sort of just capturing the first order patterns, but the neural network is capable of learning much more interesting syntax. And so we can use it as in silico oracle. So we can do lots of counterfactual predictions in silico using synthetic sequences. So for example, let's say you're interested in the spacing syntax between two motifs. You can generate random DNA. You can insert motifs, change spacing, use a model as an oracle to make predictions and see if it has learned some interesting spacing relationship. You can do this also for genomic sequences through combinatorial in silico CRISPR-like experiments. And again, a small model allows you to do this at scale, at very high throughput. And of course, then you can also use a model to do counterfactual prediction of variants, genetic variants you may have seen or you would like to query. And we can do this for common rare variants, SNVs, and short indels. All of it works pretty well. So let's jump into what these models look like. I'll explain the Chrome BPNet model. It's actually remarkably simple. It's not a complicated model. It just takes 2 KB of local sequence and maps it to 1 kilobase of base pair resolution coverage through a simple convolutional architecture. There's one convolution layer. There's 8 dilated residual blocks. They help you expand receptive field rapidly. And the model makes 2 complementary predictions. One prediction is the total coverage over the entire sequence. That's a scalar that just captures the total number of reads over the entire sequence. And the second head of the model learns to distribute that total coverage over every base pair. So it learns a probability distribution over every base pair. And these 2 loss functions are simultaneously optimized. So what we can do is then compare observed versus predicted on held out chromosomes. This is performance in peak regions for the total coverage over 1,000 base pair peaks. You can see it's pretty good, correlation of 0.84. Again, this is not replicate concordance. You shouldn't expect it because we're using only 2 KB of sequence, but it's still really high. And if you look at the base pair resolution probabilities of reads across the sequences and you compare with the observed distribution of reads, you see this is replicate concordance, the Jensen-Shannon distance between observed and predicted probability distributions. This is the distribution of JSDs. Closer to 0 is very accurate. This is the replicate concordance. This is our model versus the observed data. You can see there's pretty good overlap. And these are sort of baseline randomized profiles or the average profile across the whole genome. So the model is pretty accurate, and you can see this even at base pair resolution. If you zoom into any particular regulatory element, this is the observed data at base pair resolution. This is the predictions at base pair resolution. And like I mentioned, we can interpret the model and try to figure out which base pairs are driving activity. And in this case, the model says that there is something right here that looks like a motif. This, in fact, is a well-known motif. It's the CTCF motif, one of the most, the Chuck Norris of the genome. Everybody knows CTCF. And then there are all these other base pairs around it, which are weird. I mean, the predictions of the model are very accurate, but the base pairs don't make any biological sense. So this is for a single enhancer, but we can go across the genome and aggregate these motifs. What we realize, if you naively train this model, which appears to predict the data very accurately, it not only learns transcription factor motifs, which you expect it to learn, but it learns the TN5 enzymes sequence bias, and that actually dominates the model very significantly. So this is the first important thing to note, that if you naively train models on genomic data, any kind of biological data, and you're not careful about correcting for technical biases, you will rapidly run into this problem that the model will learn the biology and the technical biases. This is, of course, a drawback of not pre-processing the data. So we have fed in the raw data. Often people will do complicated statistical processing to remove these artifacts and then train the model on that. That's also a reasonable approach, but maybe we can do both at the same time. Let's see. So now we know the model has learned all these biases, which of course we would like to correct. So the nice thing about the human genome is you get a control experiment for free, because you have 3 billion base pairs and only about 2% of the genome actually has signal, these peaks of activity. So the rest of the genome is basically background, right? The background is directed by transcription factors. It's either going to be completely random if there's no bias at all, or it's going to be entirely directed by the TN5 enzyme sequence bias, right? So what we can do is, if we want to learn a model that only learns bias, we can train it on the background regions. There's regions that are not peaks, right? And when you do that, you actually learn an incredibly accurate model. It learns the profiles exactly accurately in background. That means sequence can predict the background profile of TN5 transposition outside regulated DNA very well as well. And if you interpret that model, you see that it learns many variants of the TN5 bias motif. So now what we have is a model that's confounded by bias, and it has bias and biology built in, and a separate model that has learned only bias. So there is a very simple trick that you can use to correct the data, which is you regress out the bias, and you fit a new model to the residual. That's exactly what we do. So you fit this model in 2 phases. You have the frozen bias model. We now use it to predict bias in peak regions, which were confounded. And then we regress that out, and we effectively fit a second model that learns to fit the residual after you remove bias. You get the same exact performance globally, but you've now routed the bias through one component of the model and hopefully the biology through the other component. So let's see what happens here. Let's go to the same enhancer. This is the same region, again, confounded by this kind of spurious bias. You can predict what the bias would look like in this region using the bias model, and you can see it actually is remarkably similar to the observed attack-seq. These spikes you see are really captured by the bias model. You can see where the protein binds, there's actually protection in the attack-seq data, whereas the bias model is predicting spiky transposition there as well. You can interpret the bias model, and you see the base pairs driving the bias are very similar to these base pairs in the flanks. But the bias model is not learning the CTCF motif, which is exactly what you would want. So once you can regress this out and look at the residual signal, it is this beautiful latent footprint which was hiding in the attack-seq data, which was confounded by the TN5 bias. So we can back out this latent footprint, and then when we interpret the bias-corrected component of the model, you can see it learns the CTCF motif and these little ZBTB7A repressors, which were in fact hidden in the original model. And just to take you back to the other approaches which actually do the bias correction in pre-processing, there have been methods that have done this before. If you fit models to the bias-corrected data from these pre-processed approaches, you can see that they, in fact, do not fully correct bias. They do partial correction, but not completely. So the key point here is that a neural network trained naively on the data, it learns biology and bias, but you can train the best possible bias model with a neural network, by simply training it on background or a control data set. You can then regress out the bias, fit the residual, and you get really beautiful bias correction, right? And once you do that and you look at the motifs across the genome, or the patterns learned across the genome, you see that the TN5 bias is completely gone, and all you learn is transcription factor motifs as you would. Now, we can actually do additional experiments to make sure that you haven't overcorrected, because you may correct for bias, but you may easily overcorrect for things. Luckily, we have two assays, DNA-seq and ATAC-seq, that both profile chromatin accessibility but use different enzymes. So the claim would be that if you have a method that corrects biases very well, after bias correction, DNA-seq and ATAC-seq should be very similar, much more similar than they were before bias correction. So this is a region in the genome. You can see the smooth ATAC-seq data. This is typically what people call peaks. At base pair resolution, this is what the data looks like. If you train a model naively on this data, and this is a held out chromosome, the predictions of the model are extremely accurate. They mimic the data exactly as you see it. Here's DNA-seq for the exact same cell type, same region. Again, the peak, the smooth peak overlaps very nicely with the ATAC-seq peak. The base pair resolution profile is completely different from the ATAC-seq, doesn't look anything like it. If you again train a naive model on it without any bias correction, it replicates the DNA-seq. If you correct the ATAC-seq bias, it predicts this bias-corrected profile, which is quite dramatically different from the observed data. You can start seeing it looks quite a bit more similar to the DNA's data. You can see the footprints aligning quite nicely. And if you do this for the DNA's data, you can again see partial correction. The DNA's enzyme has less bias than the ATAC-seq enzyme, so the correction is less dramatic. But you can see that after correction, the footprints align very nicely, and more importantly, the underlying sequence syntax the model claims is driving activity is identical from the two assays. So the model is able to exactly pull out the precise base pairs that drive activity from two assays with very different biases that originally looked very different. And this is not just for one locus. You can do this genome-wide, and you can see this kind of dramatic improvement in concordance of the DNA-seq and ATAC-seq experiments before and after bias correction. And similarly for the feature attributions, the sequence attribution scores, if you compare the DNA-seq and ATAC-seq attributions, you can see this dramatic improvement in concordance as well. So we can show how you can use neural networks to correct biases and reconcile experiments that appear to look very different originally, but are capturing the same biology. We've done this at scale now at hundreds and thousands of data sets. And of course, we want to validate this. We're calling all these motifs, these high contribution score motifs. Are they real? Is the model hallucinating this stuff? So luckily, we collaborated with Len Panacchio's group, and they did this pretty amazing block mutagenesis experiment. This is actually taking DNA sequences and block mutagenizing each piece one at a time, putting it in mouse embryos and measuring reporter activity. So you get a whole body scan of the embryo to see not only whether the enhancer is active, but also which cell type or tissue it's active in. This is an insane experiment. They've done tens of thousands of these sequences. And we've trained models in humans and mice. This is done in mouse embryos. So we can actually test the generalizability of a model trained in human cell types in the mouse. So the sequence it's a human sequence, it's put in the mouse, and you're measuring activity. So I won't show you millions of plots. You can look at the paper. There's extensive validation, but very broadly, all I will say is that even though you're training the model on chromatin accessibility and you're testing it on gene expression of reporters in mice, there's pretty good sensitivity and specificity. We're getting about 88% specificity for 60% sensitivity. And more interestingly, we can take the actual block mutagenesis results, and we can use a model to interpret why a particular block increases or decreases activity. So we get a very nice explanation. Oh, it's this cryptic motif. It's these two things happening at the same time. This mutation creates, this motif, and so forth. So we get the explanation power for free. So we've done other large-scale validations as well, but this is just one example of a pretty difficult validation experiment. Again, the readout is chromatin accessibility. The model's trained on. The effect sizes are being tested in a mouse reporter assay, which is measuring expression. So it's pretty good generalization. We can also discover novel things. So motifs, people think motifs, who cares? You can just run meme and get motifs. Well, let me tell you that the lexicon that we know about regulation in the human genome is extremely incomplete. And we will have a catalog coming out soon where we've collated neural network learned lexicons from tens of thousands of models. And I will just say that the motif landscape is non-trivial. And here's one example. FOS is a well-known transcription factor. It has this AP1 motif. TED is another transcription factor with its well-known motif. In the IMR90 fibroblast, the chrome-BPNet model for ataxia learns this composite element. It is a FOS and a TED that is exactly 6 base pairs apart. Now, usually when this happens, it's very interesting. It usually indicates a cooperative interaction. But just the presence of two motifs 6 base pairs apart doesn't tell you that something's cooperative. It just tells you that two things are 6 base pairs apart. They could happen due to many reasons. We can use a model in a counterfactual setting to test cooperativity. So how do we do that? We can take a piece of random DNA. It has no activity. You can see the black line. We can push it through the model. The model predicts flat signal. We can take the FOS-only motif, put it inside the sequence, and it predicts mild activity. We can put TED only. It predicts mild activity. You can compute the additive effect of TED and FOS, 6 base pairs apart, as if they were independent by literally adding up their signals. That's this signal right here. And then you have the joint effect of the two of them together. There's this amazing amplification. This is classic cooperativity, right? And you can also test for the spacing. Is a 6 base pair an artifact or is it real? Well, from the perspective of the model. And you can see that, again, you can play with the spacings in silico and predict effects, and you see this massive amplification only exactly at 6 base pairs. And we've done this for many other known composites as well. This is a novel composite that's never been seen before. And we haven't validated this experimentally, but we can fit another neural network to ChIP-seq data of FOS, which is one of the partners right here. And it's a completely different model trained on different data. It's ChIP-seq data, not ATAC-seq. And we can replicate the exact syntax. So you can do the same in silico experiment, and you can see the same amplification, and you can see the exact same strict spacing. So actually, a bioarchive paper just dropped today morning from my lab and Will Greenleaf's lab. We've done this at scale for a big fetal atlas of ATAC-seq data, and we've discovered hundreds of these composite elements, many of which are very novel. And the lexicon is substantially more complicated than we think. We can also use the models for interpreting genetic variation. So here's an example. This is a quantitative trait locus. It's a variant that has been shown to impact chromatin accessibility through genetic profiling and chromatin accessibility profiling. We can, again, use a model in a counterfactual setting. You take the reference sequence. It has a C allele at this position. You switch the C to a G. You see this really nice, beautiful amplification. But you can interpret the model and understand how it's doing this. And you can see that the C allele has this weak signal due to this kind of low affinity SPY1 motif. When you swap the C to a G, it creates a very strong SPY1 motif that cooperates with this other motif nearby to amplify activity. So you can get a very nice explanation for this. This is, of course, one example. We can do this at scale. So we're going to now validate against a chromatin QTL data set. This is basically ATAC-seq done in hundreds of individuals of diverse African ancestry in lymphoblastoid cell lines. So using this, you can associate genetic variation with chromatin accessibility. We identify 11,000 CA-QTLs that are in peaks. And now we have this chrome-BPNet model that's trained in a reference individual of European ancestry. So it's a different ancestry group. It's a reference individual, and we've trained it on the reference genome. We've not even seen the personal variants. We're going to try to see if we can counterfactually capture these effects. So we just simply take all these variants, we push them to the model, just as I showed you before, and we correlate the observed betas on the x-axis against the predicted betas. This is not 0.9. Again, it should not be 0.9. Actually, replicate concordance, if you do the QTL experiment twice, the correlation's about 0.8. So we're not that far from a local model that's only seeing 2 kb. And what's more important is we are very directionally concordant. So what that means is like the effect sizes are not only correlated, but when you have a positive effect, the model predicts a positive effect. When you have a negative effect, you predict a negative effect. Now let's see how this does against these large long context models. So we contrast, again, I showed you the results of a small model with 6 million parameters trained on one data set on a reference genome. The opposite end of the spectrum is a very large multitask transformer model. This is Informer from 2021. We've also benchmarked against Borzoi, which is the bigger version of this. Informer has 385 kilobase receptor field. Borzoi has 554 kilobase receptor field. This is trained on literally 30,000 tracks on human and mouse. So obviously it should be learning much better than us. But actually it ends up doing not that great. In fact, if you take the predictions from the Informer paper, the same correlations I showed you right here, and we can compute these correlations as a function of the CAQTL threshold, just so we are threshold independent, because you can always play around and cheat with your thresholds to get something that looks good. So on the x-axis is all possible thresholds. On the y-axis is correlation from the model versus the observed betas. If you take Informer's predictions from the paper, it is actually remarkably poor, which, our models are great, but that didn't make sense to us. So we went and tried to figure out what was going on, and we realized that they, for some reason, were predicting the variant effects using 100 kilobase of profile that they're predicting. So you can imagine a variant has a tiny effect at a peak, right? It destroys a peak completely. The rest of the genome stays intact. When you compute the delta, you end up swamping your own signal, right? So actually, they're much better than they think. So we recomputed them. We did them a favor. And when you do that, you actually get this nice boost in performance, which is the green triangles. But if you compare it to our models, we are still much better for the exact same match depth. And in fact, if you increase the sequencing depth of the experiment, not the size of the model, just crank up the read depth, tell your collaborators to create 10 times more reads if they can, you actually get a nice boost there as well. Okay? And this is not just for effect size prediction. You can also compare binary classification. So here we are looking at CAQTLs, right, the significant QTLs, and we're comparing the actual effect sizes. We may also want to classify, can we identify CAQTLs against background variants, right? And these are well-controlled variants. These are background variants in peak regions. So you can't cheat. They're not in background. They're actually in peaks. And the classification of a random classifier would be about, average precision would be about 0.05. So it's a pretty hard problem. Again, Informer's original scores look pretty bad. After we correct them, they are just maybe slightly better than us. But again, if you increase read depth, you get this nice boost. Okay, so model size is not everything. Read depth can actually give you a lot of power over the number of parameters in your model. We've done other such benchmarks, including with Jesse Engritz. We have this paper recently out. They have this variant flowfish experiment, which is sort of like the block mutagenesis experiment I showed, but this is in genome, CRISPR with effects on the target gene. Here we're looking at the promoter of a gene called PPIF. Now, you would have expected a model like Informer, which is trained on gene expression. It's a long context model to have learned a very good model of at least the gene's promoter. This is the predictions of Informer against the actual CRISPR experiment. It's quite low. It's about 0.42 if you use Informer's expression head. If you use Informer's chromatin head, it's actually quite a bit better, 0.62. And if you use ChromeBPNet's predictions, it's 0.65. So we are actually quite a bit better even on promoters using accessibility to predict expression effects of target genes. And the other class of large models we have are these DNA language models. And these are pretty hot right now. Many of them are absolutely massive, and they take months and months to train. And we've tested them against, we had this publication in NeurIPS called Dart Eval. It's a series of tests we suggest of trying to discover whether a DNA language model has learned very basic things like, has it learned motifs, to the more complicated tasks like counterfactual prediction. And we test these models in zero-shot, probed, and full fine-tuned, okay? And these are all the latest models. We have Evo and Evo 2 as well, and they don't perform much better than this. A random classifier would have an AU-ROC of 0.5, zero-shot, probed, fine-tuned, just slightly better than random. Here's ChromeBPNet. And the fine-tuned model sees the exact data that ChromeBPNet sees, okay? So it's not like we are seeing anything more. And these are two different QTL data sets, and we substantially outperform them. So we can do this for QTLs, but we can also go to GWAS, the first slide I showed you, and we can see if we can actually recover fine map variants from GWAS loci. So here what we're doing is on the x-axis, we're looking at posterior probability thresholds for fine mapping for coronary artery disease loci. So as you increase the posterior probability, you're getting more and more confident in terms of identifying the causal variant. This is a genetic approach to fine mapping. And on the y-axis, you're seeing the overlap enrichment of chrome BPNet's predictions against the fine map variants. And you're looking at enrichments observed as expected. You can see that as you increase the PIP threshold, you see these nice skyrocketing enrichments, and you see the enrichments improve as you increase chrome BPNet's prediction performance, as you would expect, right? And by the way, this is a model that's trained on ataxic data from smooth muscle cells, single cell ataxic data from mice, and we're using it to predict variant effects in the human genome. And interestingly, the mouse model does better than human smooth muscle cells. The reason is because we have the mouse data coming from a mouse model of atherosclerosis, where we feed it high-fat diet, and we track it over time, and we get all these unique cell states as the cells, the vasculature in the mouse is actually getting fibrotic, right? So you're getting these very unique cell states which you don't capture from the human vasculature that you just pull out from patients. So in fact, in this case, the mouse model does slightly better than the human model for fine mapping. We can interpret loci. So this is a very exciting result, and we actually see this in many loci. It's not one causal variant often. Sometimes it's two inside the same enhancer. So here we have two variants right next to each other. It is a fine map variant. The model predicts it exactly as having the effect in smooth muscle cells. The two variants right next to each other, and you can see this beautiful interplay between them. This variant on this haplotype has the A allele. The A allele creates an AP1 motif, an activator. The G allele destroys it. This other variant right next door in perfect LD, the C allele on the same haplotype, in fact, destroys a ZEB1 repressor, whereas the T allele creates the ZEB1 repressor. So on one haplotype, you're creating an activator and destroying the repressor. On the other haplotype, you're creating the repressor, destroying the activator. So you get this massive effect size. You see like the enhancer is completely off on one haplotype and highly on the other. So there's really interesting variant coordination going on. And we can also do this for rare variants. So this is an example of a patient from the DDD consortium, very severe neurodevelopmental disorder. We've trained these single cell attack models on fetal tissue, fetal brains, so all kinds of cell types from fetal brains. We are able to prioritize tens of thousands of these rare disease variants. We've not validated all of them, but this is one we did. This is in a gene desert. It's really far. It's 384 kilobases from CD83. It's 748 kilobases from JARED2. If you push it through all the different models, this specifically scores only in glutamatergic neurons. You can see it's a pretty complex enhancer with complex syntax. The A allele creates the high activity, the G allele creates low activity, and it does so by creating an NR2F1 repressor. So you have the mechanism as well. Now we take these two constructs and put it back into our mouse, again, thanks to Len Panacchio's lab, Michael Kozicki. And if you put the reference A allele, you can see activity in the brain. Specifically, you put the G allele, and you see exactly what you expect. I'll just end quickly by saying that there's an additional problem with these large kitchen sink multitask models. And that's if your goal is to actually do causal inference and design. In fact, we have very strong evidence, and we have a paper coming out soon, that they're learning pervasive features, spurious associations, which are not causal. I'll just show you one example of this. This is Enformer's predictions for a particular locus. There's a strong peak here in fibroblasts. You can just take this GRHL motif, and you can insert it in fibroblasts to see what it does. And if you insert one instance, you see some decrease in activity. If you insert two instances, you see a little bit more. If you insert 3, 4, 5, 6, you can shut down the enhancer by inserting this GRHL motif. And what's really interesting is that GRHL is in fact not expressed in fibroblasts at all. So the model has learned a spurious association. It has learned a fake repressor, which when you put into the system, it actually predicts false effects. So I'm out of time, so I won't summarize. Hopefully, my talk gives you an overview of how basically lightweight single-task models can actually perform quite well, and you can use them for interpretation. And I'll just stop there and thank my lab and my collaborators and funding sources.
let's have one question.
Hey, Anshul, very nice talk. One very common sort of epigenomic assay that I don't think you mentioned you've developed a model for is histone ChIP-seq. So, these are very widely used.
Yes.
Are there unique challenges there, or what do you think about that?
Yeah, we actually have pretty good histone models. They are corrupted with a new kind of bias. So we've been spending the last year trying to figure out how to correct that bias. We have models that work really well, but I think it's basically nucleosome positioning bias. So that's not like a technical bias, it's actually a biological bias. But if your goal is to figure out what syntax drives histone modification versus nucleosome positioning, you would want to, again, reroute that bias in a specific way. Otherwise, when you interpret the sequence, you get these haze of GC80 frequencies, which are basically nucleosome positioning signals, and you want to be able to distinguish that. So it's mostly an interpretation challenge right now, but we do have pretty good histone models. Sorry for running over time.
Okay, let's thank our speaker again.